They have left. I'm going to start the presentation then, and then when Adam comes back, I'll grab him over so he can help on the geeky side as well. So where's my presentation? There it is. I'm gonna share my screen and click on the slideshow. Now, if everything goes well, we should be able to see a very unattractive slideshow because I'm definitely not. That's not my strong side. So apologies for that. One day I'll get it right. I'll just hire somebody to do it. But we finally got to the point where we're making this Goop of ours at scale, and we're looking to pre-sell it now, so that we can have a bit of cash flow and you can have a bit of discount. So that's basically what we're trying to marry in this proposal. A lot of you have followed us. Thank you very much. There's going to be people watching this recording who probably haven't. So I'm just going to say that I'm Daniel. I'm the director of Soil Ecology Laboratory. We're down in Hampshire. And we work with compost as a bioincubator of a whole host of microbes, fungi, bacteria, protozoa, nematodes, with the aim of creating this inoculum which can help your crops grow without too many inputs. So the moonshot is that at one point our systems will replace all agrochemicals. We're not there yet. So this transitional system that we're presenting today basically hopefully is a starting point for experimentation with healthy microbes, and something that will allow you to reduce your needs, your input needs, and still have a healthy yield and have a healthy crop. So what is it in terms of our product? We really don't have a better name at the moment. It's the Goop, because that's what it looks like. It is basically a refined extract of inoculation grade compost. So we use this compost as a bioincubator when we grow fungi, bacteria, protozoa, nematodes, hundreds of species at the time. And so it's not just a select few. And then we extract them and then we remove water from it so it's shippable. And then it goes back into water very easily as a suspension. So it is a suspension, it's not a dissolved product. These are live microbes. We don't deal with dried microbes. Everything that we do is we're trying to get these microbes to be working in your soil as quickly as possible. So that's why everything is alive, nothing is dormant, which also means that it's got a shelf life, which is actually quite short. So we produce it just in time for you, and we send it on a next day basis. But we're going to get to that. Where we basically are targeting the use is your spring barley, winter wheat, pasture, other arable crops to be tested. And the reason I say that is because we've tested this on spring barley, winter wheat, and Mikey Barton actually tested this on pasture. But we would love to see other cereal crops tested, or at least other arable crops to be tested to see how far we can go with this. We have had smaller tests with horticultural crops. These are not field size; these are pot sized at the moment, on radishes, chilies, and carrots. The picture that you see here is actually the wheat trial that we had last year. On the right-hand side is biology. On the left-hand side is conventional. As you can see, the crop was quite healthy. The yield was different, though. Now, why would we look at this in the first place? So plants basically evolved with the microbial soup. When terrestrial plants started evolving about 650 million years ago, microbes had already been evolving for 1.5 billion years. And so what plants brought in photosynthesis, and so they've got a monopoly on photosynthesis. And they quickly figured out that if they feed the microbes sugars and proteins, the microbes will feed them back all of everything else that the plant needs. So from micronutrients, which would be locked in the soil profile, to even the nitrogen from the air. So if you're using the Goop, we're trying to restart that system, and so that your plant can actually request nutrients and stimulants, because stimulants are also a thing with microbes. If you use the Goop, it's, in our opinion, the fastest way to replace missing microbes from your soil's DNA pool. And if you use it with the seeds, you give your seeds a chance to work with a healthy set of microbes. We've also seen that we don't need fungicide coating when you're using this microbial product, and this really helps you speed up building the health and resiliency of your soil, so that it's less prone to disease and pests persisting in your soil. So what does it do? So we have tried to push it to its extremes, to see what you can do without any other inputs. So we've done the risky bits so that you don't have to risk as much. We have tested this on spring barley. With only 3 applications, we had a 40% increase over control, and we got to 12% to the conventional yield in one of the plots. We did the winter wheat trial. We produced again 3 applications, which produced a measly 5 tons to the hectare. But because there were no inputs, if you think about the 5 tons, that's 90 to 110 kilograms of N that's required. So the biology found 90 to 110 kilograms of N. We now recommend the Haney test, and we can help with that to see what is your soil's potential if you have the biology in it. So if you use a usual test, you might have 20 kilo. You might be showing that you've got 20 kilograms of N per hectare. But if you use the Haney test, you might be able to show that there's more. And with an intelligent use of this product of biology, you should be able to unlock more nitrogen than you normally would. Pasture. This is a client trial. That was an interesting one. There was a comparison between the Goop, 50 kilograms of N, seaweed extract, and some controls. In general, across all of the different plots of preparation of the soil, the biomass was the highest on the 50 kilograms of N, then the Goop, then the seaweed extract, and the control. So it was trailing that 50 kilograms of N, but then there was an outlier. In the field where the ground was sprayed off with glyphosate and reseeded, the Goop outperformed everything by 24%. So that's another idea for an experiment, because we're all using glyphosate. Much of us are using glyphosate to prepare a seedbed, but then that could have a negative effect on your crop. And so replenishing that healthy biology after the glyphosate is something to do more trials with. We did smaller scale trials. Again, radishes, we had 22 to 55% increase over control. Carrots, 19% increase over control. Chilies, 47% increase over control. And I'm going to speak about those individually now. So the spring barley, this is what it looked like. Half a hectare total area. There were 2 controlled replicates and within a conventional field. So the strips on either side of this trial were basically treated as if those are our conventional applications. And this graph basically shows you in tons to the hectare what the result was. And the biology basically outperformed the control by 40%. And it's significant because a lot of people treat compost. We use compost as this bioincubator, right? A lot of people treat compost as a replacement of part of your N. That is an established procedure, that is established practice. People will make compost, will spread it on their field, and the NRMs, the land crops of the world will tell them, this is, with that amount of compost added, this is how much nitrogen you're adding, and you can basically cut out your synthetic nitrogen by that amount. Now, here's the thing. This field received an inch of compost that was produced here on the green waste site. So we're actually located on a 160 hectare farm, which is arable, conventional. This is where all these trials were done. And it has got its own green waste facility. It processes 5 to 10,000 tons of green waste per year that come from landscapers. And then they don't sell it largely, it just goes onto the fields. And that is on that calculation, that is the maximum amount basically the fields can get are based on the calculation of how much nitrogen is available. So this field received an inch of compost that got plowed in, and still our 3 applications of our biology outperformed the control by 40%. So in terms of how this happened, we've got our hypotheses. We've got the Manchester University ready, waiting for that grant money so they can actually answer those questions. But in practice, we've seen that adding this biology actually unlocks more nitrogen than you would have normally, even if you're adding compost at scale. The application rate was about 10 liters of our Goop per hectare per application, and we had 3 applications, and they were done in April, May, and June. Now, in terms of how to apply things, it would be best if it was applied in wet conditions. This is like opposite to what you would be doing with chemicals, because the microbes need to survive. And I'm just going to there we go. And then there are now practitioners out there who do their applications at night. We didn't do any of those things, and we still had this result. So whether it was the survivability of the microbes or it was foliar feeding by the extracts, we don't have that answer, but we have the result. If you were to take the cost that we're presenting, the total cost for this application was 120 pounds per hectare of the Goop to get to those roughly 4 tons. In terms of the winter wheat, with our measly 5 tons, this was done on 5 hectares, and 4 3/4 were in biology, and 1/4 was left as a control. This was not a replicated trial. This was us trying to push the size. Because we're a small company, we do everything by hand. We were trying to see how far we can push this at our current scale. And so we got to these 5 tons, but what's interesting, again, 3 applications, 14 liters to the hectare. What's interesting, Adam collected these heads close to harvest, about probably a month before harvest, at random, weighed them up, and got to these results where the heads on the biology side were the heaviest of the lot. We didn't do any numbers in terms of tillers or numbers of plants per square meter. So obviously the yield came from the other 2, most likely. But this is an interesting observation that we found. There was, again, no other inputs. The Goop cost would have been 168 pounds, and at feed wheat price of 177 right now, that would have been 885 pounds revenue, plus application, obviously, costs. There was nothing else used. There was no herbicides, and we had a volunteer oilseed rape, first of all. And then the other thing that we had was cleavers and groundsel as our weed pressure. So we probably lost about 2 tons due to weed pressure. But we wanted to test everything when it comes to this Goop. So again, so you don't have to. So we know that at least in year 1, you will have weed pressure if you don't have anything else. Now, with that pasture trial, where basically after glyphosate use, it seems that the Goop actually helped with the bounce back of the plants. We would like to see more trials where basically there is obviously the herbicide used and then our Goop is applied afterwards to see what happens in terms of the crop. In terms of instances of disease or pests, we saw no instances of pests, and there were rust instances, but we didn't use any fungicides, and we had rust in both the conventional side and the biology side, and the biology side was holding up quite well to it. In terms of that pasture trial, the trial was done by Mikey Barton. He's with us today at Matt Benson's farm in Scotland. And this is on a 1 hectare square where there were preparations. Vertically, you see different preparations of the soil, and horizontally, you see different applications. So this was done in the heat of the summer. This picture was taken in the heat of the summer last year. And you can kind of see that the goop in the bottom is actually holding up the best in the drought. And that was our main observation from this trial last year, that the plants were actually holding up quite well, even though we had record drought last year. So this was a single application in April, I believe, but it was a single application in the spring, which would have made the goop cost 56 pounds for that application per hectare. As I mentioned before, basically we've seen that improved drought resistance, and also in this little square here, in the bottom right corner, this is where it was sprayed off and drilled, and this is basically where the biomass was the highest in this strip. What's interesting, and this is a conversation that we hope to have with Mikey in the coming weeks, and record that for you. They are now collecting second-year observations, and that's important to us because they didn't have any more inputs on any of these fields apart from grazing. So grazing was the only input, if I remember right. Mikey, you can correct me. And we did other things. Again, it's smaller scale. The sand tomatoes on the left, this was me being cheeky. I don't grow things. I'm terrible at growing things. I stick to decomposition. And I bought play sand from B&Q, and I filled the play sand with different amounts of our goop. And in this picture, you can see the difference between 100% our goop mixed with water, obviously, and just water. It's a 350% difference. You can see that the plants actually had something to work with. And I've got a repeat of that outside, and the tomatoes are nice and dark green, the leaves. So we'll see what happens with them. And then the radishes that were done, this particular first trial was done by a colleague of ours, Silke, and she only put 3 milliliters of goop into 25-liter planters filled with Sutton compost that she purchased online. And she had a 55% increase in the weight of the radishes itself, not the whole biomass, but the radishes. We replicated that in control conditions, and we got a 22% increase over control. And then the chilies, they're also very interesting. This is what Adam did in his conservatory. Adam is our main geek. Adam, will you be joining us? Or are you doing something else? Cool. When he finishes, he's going to join us as well. So the chilies are interesting because Adam had 8 pots, so it's a good, decent amount of replicates, and all 8 pots were fed. All 8 pots were fed amino acids. All eight pots had micronutrients added. All 8 pots had the same compost that we used to extract the goop out of. And the only difference was that the little seedling pots, when Adam first put the seed into those seedling pots, he watered 4 of these with the goop, with our compost extract, basically. And the difference here in the yield is 47% dry weight. Now, when we presented this to Professor Duncan Cameron from Manchester University, his response was, you've created some legacy effects with your compost extract, and we don't know what it is. We'd love to see what it is, because at the moment, the only thing that we use in the industry is actually something that we fear that is ending up in seeds, and people are eating it, and we don't want it because these are persistent chemicals. So nice pat on the back, because we've done something that that the industry is doing with scary chemicals. So the pre-sale is open now. What we're trying to do is we're trying to get this into your hands so that you can experiment with this, but risk as little as possible. So we are doing this as a pre-sale. We have been selling the Goop in small quantities for a year and a half now on our other website called Soulsmiths. And we've been selling that in small quantities at £100 a litre. And we knew that this is a ridiculous price. We have to go down to an economical cost for farmers. So so this batch is at £5 a litre. And as a thank you for participating in this early, because this is in production right now, we've lowered that down to £4 a litre. And for the first sort of 20 hectares worth, and I'll tell you why this is £336 for a hectare, but I'll tell you what in a minute. For the first sort of 20 hectares worth, we're also doing a free shipping version so that you can call it off whenever you need it, and we're going to cover the shipping. Now, we need to sort of have a good chat, probably individually, so you can give us a better idea of how you're going to use it. Because our assumption is that you would have an opportunity of up to 6 applications in a growing season. 1 application to the soil, 1 application to the seed, and then up to 4 applications overhead. And this is where the 6 applications come from. This is where this cost comes from per hectare. Now, all of our trials were done up to 3 applications, nothing more than that. So could you do a trial of 2 hectares with 3 applications? Would you be interested in doing a trial on pasture where you're just doing that 1 application like Mikey Barton did? Could you do, just an application to the seeds, which is actually the most economic, but on a larger area? And then you're using this as part of your system where you're trying to reduce your inputs. So we're open to suggestions on how to do this, and we'd like to basically to get this into your hands and see how how you're going to use this to your advantage. We believe that this definitely is already in a format that can unlock at least part of the nutrients that your plants can get to, so that you can use less of your inputs. How we're going to use that's another story. We've got a list of potential experiments to do that. We don't want to pull wool over your eyes. This is an experimental product, but this is something that will allow you to experiment with biology without spending 2 years practicing making your compost. So instead of investing lots and lots of time in growing out your compost, we've gone through over 50 iterations over the last 3 years. And so we've put in quite a lot of work into mastering how to grow out this biology. And we continue this development. We need obviously funds to continue this development. So we're constantly fighting for grants. We're constantly fighting for investment. And we're hoping that with your support, with those early purchases, we can also convince more investors that there are people interested in this methodology, and that this is a worthwhile endeavor. Now, before Adam joins us with some more stuff, Mikey Barton says that's correct. Okay, fine. We can actually start with some questions. So any initial thoughts? Go right ahead.
I don't know how I get on the screen or anything, Daniel. Am I there now? Can you hear me?
Yes, we can hear you. We can see you.
Okay, right. Well, the little project that I've got thinking about going in September is I've got some pasture land that I purchased in about 2008, so it's had nothing on it since 2008. Had a few cattle, sheep, occasionally some horses on there, and we're just bringing it back into some sort of production. I've got my neighbour's cattle on there at the moment, so we've got some Aberdeen Angus, mowing it down a little bit. The idea that there's loads of different types of grass in there. It's a permanent pasture. Obviously, it's been there for donkey's years. It's an old dairy farm, so it's probably always been pasture as far as you can go back. The idea is to get it down as low as possible sometime in the autumn, and the initial idea was just to put some white clover in, or possibly some red clover with it, just to get legumes in there producing the nitrogen. And then I turned around and said, well, why don't we do a proper herbal lay in there and look at getting some worming for the cattle, some deeper rooting plants in there to take over from the nettles, etc., and the thistles that we're taking out. So looking at getting something like the Goop in there, just to get some soil biology back in. I did have some tests done, and Perry, and I can't remember his second name. Hallenby, did the test, and we can have a look. I'd like to have a look at those with you at some point. Possibly you can, because maybe we can look at using that and what comes out afterwards if we did put the Goop on, to have a little bit of a comparison. But I'd be interested in what you had to say about where we might go with applications, how to apply it, rates, probably about a 4 acre site, possibly 5 acres. And yeah, just what your thoughts would be on going on with that.
Okay, so you were breaking up quite a bit there, but we did I did hear that you're looking at about 4 to 5 acres to do anything at the moment in terms of testing.
Did you get that?
Yes, I got that. Basically, you're looking basically at about 4 to 5 acres, but you were breaking up quite a bit. I don't know if it was just for us or anybody else.
I think it's my connection. I can't. Yeah. Yeah, permanent pasture. Once turn it into and look at direct drilling.
Can somebody else confirm that this is is Mark's connection or our connection that Mark?
I think it's Mark.
4 to 5 acres.
Okay.
Mark, can we turn your video off, Mark?
Lay.
Do you want to try to turn off your video? See if we can hear you better. There we go. Right. Let's try again. 4 to 5.
Is that coming through at all?
Oh, this is coming through much better. Thank you.
No. If that's not working, then take a question somewhere else, Daniel.
That's fine. I can hear you now. If you want to say that last bit.
Hello? You good? Can you hear me? Yeah. Is that video off? Is that?
The video is off.
I pressed something that said stop video. Can you?
We can hear you.
Okay, right. Okay. So I've got some grassland. It's been grassland, permanent pasture for donkey's years. We're looking at putting a herbal lay in there this autumn. Direct types of red clovers, but I'm interested in how to in how to apply the goop, when you would apply it, rates, etc., and what I'd need to do. I presume a sprayer, nozzle sizes, anything like that. That's just I'm just trying to work it out in my head how I'd go about the job if I wanted to apply goop.
If you're starting off if you're starting off with just seed coating, if you're doing stitching into that field with small seeds, the application rate of goop is ridiculously low. And Tom actually, who's with us today, he recently applied, I believe it was 100 kilograms of seeds to 10 acres, and he used 250 milliliters of goop. So if you're doing just stitching into pasture, and using the seed coating as your method, then you can use it with a tiny amount of goop, and you can start whenever there's rain, basically. Even if you buy an amount on Soulsmiths for the high price now, it's still not going to be enormous. So then when you're following this up with spray applications like Mikey did, then we would advise the full application rate, where basically you're using up to 14 liters per hectare. You can try less, you can try more. We've never actually pinpointed what is the amount that works best. So we have basically settled so far on this 14 liters to the hectare when it's applied, and that comes out to about 1.4 milliliters per square meter, basically, of goop itself, right? And that's diluted into water to put into your sprayer. So those are the two application rates that we will be talking about.
Okay, so stitching in, is that the same as direct drilling?
Yes.
And oh, lovely. How do you go about coating the seed to put it in the direct drill then?
Tom, can you hear us? Are you able to fill that in?
Yeah, do you want me to go through what I did?
Go for it.
Please.
So, with Daniel's help, we worked out some maths on how much goop we required, and I'm just trying to bring up the document. But basically, we 500 mil of goop, and we reseeded 23 acres with it, which was a cost of £3.50 per acre. And what we actually did was reduce the seed rate by ½ kilo an acre, so it paid for itself. The way we put the goop on was we mixed it. We had the seed in 25 kilo bags, and we put 125 kilo bag in a cement mixer with I can't remember the actual. I can get the rates, but I can't find the document now. And like literally, I think it was like 23 mil of goop made it up to a pint of water. We started originally with 1.5 liters, and it was just way too much liquid. And then mix it around in the cement mixer, and until it was dry, put it back in the bag, put it in the drill. We did that 3 or 4 weeks ago. We thought the original plan was to broadcast it. Here we go. The original plan was to broadcast it, but there wasn't enough soil moisture, so we decided to drill, and there was moisture when we drilled, and a lot of it has germinated. And I put a small amount in a cup in the kitchen so I could like monitor moisture levels as well. And the fungal content on that is massive, but the field stuff's struggling like mad now. I got no moisture for about 10 inches down, so it's not a fair trial on the goop. There's also a 2-acre control on that, so I can have a look in the future to see if there is any
So
any
literally,
sort of effect.
this would be something that you could be doing on tens of acres, basically at a very low cost. You know, if you wanted to have a bigger trial, if you're just using stitching basically in pasture or, you know, reseeding. So that's basically, you know, I can't remember if you mentioned the amount of water, but the amount of water was 6.5 liters for 100 kilograms of seeds.
That turned out to be way too much. We had to pull it right back.
Okay, so but basically, so yeah, so there's an estimate of how much you can use.
So you're mixing the goop with water, and then that is being taken up by the seed, absorbed by the seed. Yeah.
Yep.
Yeah. Okay, I understand what's happening now. That's all.
Exactly.
Is there any benefit, or would you think there would be any benefit in applying goop to the spray to the field before you direct drill?
Great question, and it just creates more variables. So, you know, this is literally now a separate trial. You know, it's like because we really don't know what's going to be the end process. That's going to be the best. And in an ideal world scenario, right, you would be injecting the goop mixed with water into the soil profile so that you can get rid of any compaction that you've got, or at least give the microbes a chance to establish, you know, in the crevices of the soil. Then you would be coating the seeds, and then you will be following up with it, you know, as the plants are growing. That's basically the ideal scenario. And, you know, we are very open to all sorts of mixes. Again, Mikey started on pasture with a single application. Tom has taken the Goop for direct drilling. We've not had a chance yet, for instance, to inject into the profile. So there's a number of ways to look at it.
Sorry, the chap that was just explaining what he did to the seeds, and sorry, I can't remember his name. I was listening too much.
Tom.
Tom, is that an experiment that was started earlier this year then?
Yeah, we literally 3 or 4 weeks ago.
Yeah, no, so that's I just want a clarification on that. Yeah, because it's dry now. That's the way your problems are lying at the moment. Lovely.
Correct.
Okay. What about nozzle sizes, etc., for spraying it onto the soil? And are we talking then about spraying onto the crop at a later date? And then what I'm trying to work out is we've got potentially live invertebrates, etc., that you're spraying onto foliage that should be living in the soil. Yeah.
Yep.
So
not invertebrate, or I don't think
my biology might be slightly wrong there, but your nematodes, etc.
Yep.
Should be in the soil, yeah, and we're spraying them onto once we got some establishment or things start growing a bit more, they're going onto the leaf effectively, are they? Or
true. So obviously you're going to have some spray onto the soil, and this is why I mentioned during the presentation that you know, ideally you would be waiting for some wetter conditions, or even doing this in early, you know, wee morning hours, so that you've got some moisture there. But again, all of our trials were done in the drought. This is why in the dry, not in the drought, which is why you know, I am very open in saying that we don't know yet how much of the effect is done with biology in the long term, or with the biology dying on impact and providing nutrition to the plant in the moment. And I think there's a bit of both. And the more applications you do, the better chance you have with the biology for the biology to establish. And obviously, if you're doing this to the soil, if you're doing this to the soil, to the seed, then you've got a better chance of establishing the biology. And then when you're spraying it, in the conditions that we did, for instance, the spring barley, the May weather and the June weather that we did the applications were actually quite hot. And so, that prompted me, for instance, to do an experiment where, we had a tea brewing in the other room in a little jar. If you were to put that under the microscope, it was just buzzing with flagellates, lots and lots of flagellates. So I just took 3 drops of that and put it on a slide and left it overnight on the windowsill. Came back the next day, everything was fully dry. Looked at that in the microscope. There was nothing moving, obviously, because it was dry. I re-wetted it, put a cover slip on it, left for an hour, and one ciliate woke up, nothing else. So the thing is that there's definitely an effect of how you're applying this, for sure. Now, David basically was asking about how long will the Goop keep, and the thing is that we can send this on a next day basis whenever you need it. So that's something that we do. And what Judith has pitched in. If you use compost and you do your own extraction, then you can keep the compost for longer, obviously, and you just have to deal with your own extraction. The Goop is a convenience product, so it's something that basically is ready to use. So we basically have designed it so that we can send it on a next day basis. We've been doing this for 1.5 years now, and we've only had a handful, literally less than fingers on my hand, numbers of times where we had issues with shipment. So, and we've had over 100 clients for this product now. So we don't have an issue with shipping the product, and people use it and come back to us with positive results. So we know that it works in that system.
Can I go through a couple of questions?
Yes.
Probably have to talk about afterwards a bit, but I think it's definitely got legs. But I just think there's some more data to be pulled out of the trials, and the trials need to be increased or increased in size. So a few questions on you had the cost of the FERT plan, your sort of the Goop FERT plan, but not the cost of the conventional FERT plan. So I would really like to have seen gross margins and net profits for the 2 crops. So using the Goop, the net margin was X, using the conventional was X, Y, and the control was Z. So you can actually say, well, these farmers aren't just farming for yield, it's more profit.
I can't remember off the top of my head. It's a fair question, so I can fill in a little bit right now, straight away. So I can't remember off the top of my head the amount of N used on the spring barley. Obviously, the value of the yield was there. But I can remember actually what was used on winter wheat. So winter wheat got 11.4 tons of yield on conventional side, and we calculated 210 kilograms of N, plus other inputs. So we do have a full sheet actually of what was used. So if you were to compare it to the actual cost that we had, was definitely not at that scale, was not comparable at all. So it definitely warrants more trials. That's why we want to basically put it into people's hands now, so we can do these 1, 2, 3 hectare trials across a number of soils across the country, across a number of crops across the country, across a number of approaches, so that we can have more of these data points. Now, we do have a bit of an offer for that. Once we are selling this, so you take part in the risk. However, we're still very much hopeful that our investors will come through. With your purchases, it's going to be easier for us to get that investment. And that basically means that once we have the proper investment, we can return the cost of the purchase after we receive the data from you. So if you do a proper trial for us and gather as much data as possible, we will return the cost of the purchase of the Goop. How's that?
Sounds good. Next question. How are you applying the Goop, and why have you chosen that method?
Whatever we had available. So here on the farm, the only thing that we really had available was an overhead sprayer. So Chafer was our poison, and we just had it rinsed off because it's used for everything. So we asked the contractor to rinse it out, and then basically we made the extract. We got the chafer pump to bring it into the tank. Off it pops, and we did it 3 times in the season. That was the only thing we've used. With the wheat, the only difference that we had, from the spring barley is that we wanted to put it into the soil somehow. So we had this volunteer all-seed rape because we didn't use any glyphosate. And so, I do have a picture somewhere where basically half of the field was glyphosated from the all-seed rape and was completely dead, and half was lush green because of it growing. And we sprayed that, and 3 hours later it was harrowed in, and that was the first application. So that was our closest application to closest method to actual application to the soil, to the profile of the soil. It's interesting that what we found was 2 or 3 weeks later, we basically went and sampled the soil, and we found that with that method of harrowing the oilseed rape, the population of nematodes after our application increased 200-fold, 200 times. We tested again because we couldn't believe it. First, we came up with 600 times number. So we tested several times, and we got to 200 times increase in the population of bacterial feeding nematodes. And we attribute that to that extra food from the decomposing oilseed rape. So there's a lesson there, I think, that we could potentially follow. And then there was an application to soil right after drilling. And then there was a spring application in around March time, the first time the machines could come out. That was everything on the wheat, nothing else. And then the spring barley had fungicide coated seeds. We applied several days after emergence. That was the first application. And then 2 others where the crop was growing. And everything was just a sprayer. In terms of pasture, that was a sprayer as well. All that Mikey did was a sprayer. So yeah, we're definitely open to other tests. Phil invested, for instance, Phil is here with us. He invested in a piece of equipment where he's got the ability to rip and drip. So there's something to play with there. There are some other people that we know that have also invested in that sort of kit. So we might look at doing trials with that. There are people who already have peristaltic pumps on their drills, so they can simply drip into the furrow as they're drilling, so they don't even have to coat the seeds. So they can increase the amount of microbes that are actually going there. You can mix it with other things. There's a whole host of ideas for Adam actually produced a whole host of ideas for testing. Let me just try and pull this up because bear with me. I'm going to share my screen. I'm going to allow Adam to talk over this maybe a bit because this is interesting. Some of this stuff is for us only, and some of this stuff is for you guys to play with these ideas. Adam, do you want to maybe talk through some of the ideas that we've got to test the Goop with?
So it's basically incorporating different nutrients into the Goop. So as you're delivering your biological inoculant, you can also deliver some chemistry with it. If you check out our chat with Tim Parton about growing oilseed rape with no pesticides, he talks about delivering mineral nutrition into the furrow. So that's one thing we're thinking of incorporating into the Goop. There's already a lot of mineral nutrition in it. It displays the same kind of cation ratios as how the whole ecological agronomy movement of the States would call the ideal soil, right? So calcium is between 60 and 70% of the base saturation. Magnesium is between 10 and 20%. Potassium usually hovers around 5 to 7%, and then about 1 to 2% sodium. So it's kind of like ideal soil, right? So if you turn that into a seed dressing, what we're doing is trying to coat the seed in ideal soil. That not only includes biology, but also the mineral amendments. So what we're starting to see with sap analysis is a degradation, particularly, soils that have, they've been farmed for a long time. They might not be as good as they once were. They take longer to heat up. So in spring, they're less responsive. The microbes take a long time to warm up, and then not until the soil warms up do the plants start to get what they need. So it would be advantageous to be able to put the mineral nutrition that's lacking in those early moments of the plant's life early on in the season in the furrow. So some of the things we're looking at is protein, nitrogen or amino acid nitrogen, boron and molybdenum may be really important just because molybdenum doesn't seem to be around or accessible during those early moments of the plant's life. Sulfur is another important one for making the plants more resilient to pest and disease. So is copper, calcium lignosulfonate we're looking into because it's like calcium sulfate, so it's just like using liquid gypsum, but it has lignin attached to the sulfur or the sulfate. So it has a fungal food. So we're putting that in the furrow along with the fungi to give the fungi fungal food and also supply the plants with sulfur and calcium. Usually we see on the saturated paste test or the SAP test that early on in the season, again, calcium isn't so mobile in the soil. And as the soil warms up, calcium becomes more mobile. And then other inoculants, like more targeted inoculants like mycorrhizal inoculants, IPM inoculants like Bavaria bassiana and Metarhizium, bacillus species, endophytes, and entomopathogenic nematodes that might be used to target stuff like flea beetles or slugs. If you've got a big problem with slugs as the plants emerge from the furrow, it might be advantageous to incorporate entomopathogenic nematodes. So lots of different things to experiment with. So there will be Goop version 2.0 and 3.0 and 4.0 and regular updates.
This is constant work in progress. We are definitely we've always said it, we present how it is. We give you, with this iteration of the product, we've tested this far. We've tested it naked. There was no other inputs apart from the chili trial. I believe the chili trial actually had lots of inputs to it, as I mentioned before, but all of the other inputs were completely missing from the mix. So it was just the Goop, it was just the biology from the compost. So what we're showing here is that you're able to save yourselves years of mastering a skill. We look at this as the missing element or a puzzle piece in the regenerative agriculture picture. So the biology is the most important bit in our opinion, and what we're trying to do is we're trying to give you a shortcut to it so that you can experiment with it without spending so much time as we did, actually. So we've put in the amount of work. Adam's hands are the most responsible for the 50 piles of compost that we've gone through. So there's a lot of experience with us, when it comes to actually practicing this.
I also forgot to mention the thing I was looking at yesterday, which is the increase in known microbes that deal with glyphosate, that cleave glyphosate into its constituents. So we've seen 175% increase in the 4x4 area we were growing our wheat for our trial in just a grow tent, like a growth chamber. 175% increase in Solicoccus terricola, which is a yeast known to cleave glyphosate, and the control increased in 29%. So significantly greater increases in microbes that deal with glyphosate if you're working with conventional farmers or a conventional farmer that uses glyphosate. I think that could be super useful.
Now, so what Adam is also referring to, I don't know if you might want to pull up one of those reports just to show an example of it. We started working with biome makers who are doing DNA analysis. I'm going to stop sharing so that Adam will be able to share.
Yeah.
So we started working with biome makers. In general, what we, in our approach in this whole route of development, we are trying to have practical tools, use practical tools as practical as possible, something that you can use even to have the most insight into what is happening in this process. So we're trying to have the full nutritional story to the plant and an understanding of the actors in that story. So we started off with microscopes, and we've been obviously training you guys as well. Some of you have gone through our training on how to use the microscope. So we've used the microscope. It gives us morphology. We can have biomass of fungi, bacteria, numbers of nematodes, numbers of. Thank you, Tom. Thank you very much for coming. We'll speak later, I'm sure. So you can have the biomass of the microbes. And then that's not everything, right? So we need to understand, well, where are the nutrients in the plant? So we use sap analysis now to understand what's in the plant, what's actually ending up in the plant. Okay, great. Well, where did it come from? So we use Logan labs because they use the Albrecht methods, which are actually something that Novacrop that does a sap analysis, they also understand these numbers. So we use Logan labs to look at the soil in terms of its mineral composition, what's available in the mineral composition. Then we looked at, then we started using the Haney test to have that available nitrogen with biology, to have a better understanding of what nitrogen we can actually unlock with biology. And then we wanted to have a better understanding of, the key actors in this whole story, because if the story is that the plants feed the microbiome and the microbiome then responds with all the nutrients that the plant needs, if the SAP analysis is showing that something's missing, then we can look at the actors and say, well, is the whole cast there? If the whole cast is there, then we need to ask the question, who's not doing their job? Who's not actually come to the rehearsal? So we started looking at DNA analysis, and we've chosen as our current partner is biomakers, because not only they give us a list of microbes, like all of the other services, but they also started looking at functions. So they can read out of the genetic material. They can read what sort of functions the microbes are actually performing, and we're trying to work with them deeper to actually have a categorized list of these microbes. Do these functions, this, that, and the other. Adam, if you're ready now, I can stop talking. You can actually share your screen. But we have now these 5 different viewpoints to this whole story. So that's and we're trying to have to build a picture that that gives us everything that we need, so that we can actually have some more control and speed up the development of these processes. So what you're looking right now at is the back end that we have access to of the platform that biomakers provide.
Yeah, so this is kind of a visual representation of the data. A lot of other companies that provide metagenomic analysis give you a list, and then I have to read that list and go through with Google and look at the potential functions of everything. But biomakers present it like this in the Explorer, which is really cool because it engages pattern recognition a little bit. I can look at the big circles and I can see who's around. Right, we've got Mortierella around. We've got lots of different fungi around. We found that this fungi seems to be showing up a lot. And if you look into the scientific literature, this is the guy that deals with glyphosate, right? So it makes sense that he would be in a conventional field where glyphosate gets used. So we were looking for increases or decreases in the population of this microbe. And then we're going to measure glyphosate. So we did a baseline study, and then we're going to do a follow-up test to see if we've reduced the amount of residual glyphosate in the soil. And then look at the increases or decreases in the instances of this particular yeast in the biome. And there is lots of stuff to do like this, right? So you might see this blue circle. I don't know if it's, it's Pythium, right? So the microbe associated with damping off. So, lots of seeds get lost every year to Pythium. We found by looking at the biome of native wheat plants in Israel, where they evolved, that's emmer wheat. We looked at the biome in the rhizosphere, and we found that usually it's dominated by types of fungi that produce antibiotics, like Penicillium, Aspergillus, Mortierella is another one. And wherever these microbes become dominant, the instances of Pythium seem to be reduced. So wherever we see big circles for Penicillium, we see really small circles for Pythium. And if you look into Penicillium, you know, the genus, different species in the genus are known to produce hundreds of different antiviral, antibacterial, antifungal compounds. And they're also endophytes. They're most likely endophytes in wheat. So it makes sense that a saprotrophic or symbiotrophic fungi that lives in the wheat biome would select against Pythium, you know, damping off of the seeds of its host plant, because obviously the fungi wants to support its host plant and wants to protect it. So super cool, right? We're seeing the form and function of native wheat, and then we're comparing that to different agroecologies to see, you know, is it behaving the same, or is it being disturbed by some of our management practices? So we did 4 samples on the beds, or we did 6 samples on the beds, and then we made 6 applications to those beds, and we did another 6 samples, so 3 for the control, 3 for the treatment. And I'm just going through and seeing changes in the biome now. Obviously, this is a really quick study. You know, it's not even a year because we're in a growth chamber. So it's literally months. But it enables us to keep running experiments, right, when it's winter. So we can learn faster than we could if we were sticking to the whole annual flow of things. We're
really, you know, working hard to get funding, you know, externally, so we can really speed up the understanding, so that you can start using this in practice, right? For, you know, with your participation in the practical trials, you know, we can definitely increase that interest from the funders, because the funders are definitely interested in projects where farmers are actually participating in this, right? So farmers are actually putting up some of their time or capital on the line to look into this. If there is interest, then there's funding. So that's what we're hoping that you can help us with. There is definitely a legislative push towards this sort of agriculture, and so if you are to invest in this product, then you are actually aligning yourself with these legislative pushes as well. Thank you, Phil. You're going as well. There's a question here. Hold on 1 second. Judith is going. See you later, Judith. Thank you. Clyde, you're asking, what were the qualities of the crops like? Nitrogen, starch. Do you want to unmute and say something more about this question? Something's wrong. I can't hear you. Okay. The qualities of the crops like nitrogen. This is basically the biggest thing. Maybe I'm going to just answer this first, because this is the biggest thing that basically is missing. This is why we say that this is not everything. If you actually, when you actually read through the listing, we actually say that the 2 biggest things that this thing doesn't do, it doesn't replace your nitrogen, and it doesn't replace your herbicide applications. In nitrogen, consistently so far, we've seen that the protein is low. Wheat, barley, pasture. We've seen that. So there is a need for extra nitrogen in that transitional period. There's nothing that this product is providing in enough. But we believe that with a proper program, you should be able to reduce your nitrogen input even in year 1. We don't know what the proper program is. This is where we rely on your experience as farmers as well, so we can design this to your advantage. But we have seen that basically that the protein count is low. It's helpful with spring barley, because the growers.
It might be more accurate to say it's the same as the conventional and control in our trials, right? So if we applied it to An organic farm like James's farm, where he gets high protein, you're going to get high protein. You apply it to a field where you get low protein, it's not going to systemically change that in year one.
So it's something that definitely in other ways of looking at it is the same. There's really not much.
And I think it might be cool to couple it with a Haney test to try and get some kind of understanding of different pools of nitrogen in the soil. So not just ionic ammonium and nitrate, but also water-soluble organic nitrogen, and then also water-soluble organic carbon, see what kind of microbial foods are around for them to use. Because it's likely that this stuff is not going to work in environments where microbial foods are lacking. And if those microbial foods are lacking and you can identify that, then at least we can try to supply them along with the extracts to optimize the application. Obviously, every soil type is different. So a little bit of investigation into that, I think, goes a long way.
Yeah. So we do have a full suite of these tests that we can help with dishing, samples to other labs. So we can help with that. So talk to us. We haven't got that on the website yet, but we'll put it on the website so it's easier for you to find. But as Adam said, we're constantly working on what else we could actually mix into the Goop so that it's a more complete product, not just a biology shot in the arm, but also something that will support the biology. It's a big question always, how to make sure that this biology survives. Anything else for the starters? Adam, did you have any more thoughts about sort of how we're actually approaching this whole thing?
Nothing more to add, really.
It's interesting from the DNA stuff. That little trial in the tent that we've been conducting, it's not a complete trial. We had to terminate it early. But the DNA analysis actually has shown that's already some needles have been moved by a few applications of the Goop. So it's something that that is giving us again more evidence that that we're going in the right direction.
Yeah, like I said previously, if you use glyphosate, then applying it after the glyphosate to try and break it down faster is a good idea. And then also maybe after, if you use insecticides or fungicides, then applying Goop after that, along with L-isomer amino acids, may help to reduce stress in the plants and also clean up the fungicide and also repopulate the fungi that you may have, destroyed by, applying the fungicide to the target organism. So
definitely talk to us about other inputs. Adam and Paula especially, they've got very proficient in understanding what is available on the market that will work better with biology than some other things with a crop. So definitely ask questions in that regard. We can help with that too. But I encourage you to look at the listing. I encourage you to give us feedback, but I definitely encourage you to make a purchase. Again, with your support, we're able to get investment and grants because then the people with money actually see that there is interest, and then we can fund this quicker, we can develop this quicker, and we can get to a very good working solution quicker. As Adam said, this is Goop 1.0, maybe 1.5, I'd say, but Goop 2.0 is around the corner, and Goop 3.0 and 4.0 is constantly in the back of our heads on how to make this better. We've got, just to give you a glimpse of that, that list of experiments, something that we can do right now is a really a potential for a very easy win for this product and for you in terms of how this could be working quicker, because our aim is always to make this work quicker. The biology needs to work with your plants. The question is how quickly you can make it work. And that's the challenge. Is it working over 5 years' time, or is it working in 1 season? If we can bring it down to 1 season, it's a no-brainer for anybody. At the moment, if anybody is going regenerative, they have to invest the time into making their compost. They have to basically have built all that skill set, and then basically they can experiment with it. So we've cut that back. So now you can start with experimenting because it's a ready-made product. And now we need to improve on that so that we can get it to work as fast as possible. And you know, another thing is what's about that whole nitrogen addition, right? What can we do about nitrogen fixation in the soil? We've been experimenting with that as well, and we've got a number of threads that are open, and we need the funding to be able to pay the hands that actually are doing these experiments, because it's quite possible that there's a, you know, with the number of hypotheses that we've got, we could potentially create more products that will help speed up the establishment of nitrogen fixation in your soil. And that means that before you even put a crop in, you should be able to stimulate the natural nitrogen fixation in your soil. And so that there's a pool of nitrogen that is in an organic format, in a little microfertilizer bag format, that your plant can access when it's time for it to go in. So there's plenty of for us to do in this development run, but Goop 1.5, 1.0 is ready for you to use, and we will start shipping end of September. So I'll show you. Actually, I'll just basically I think I've got it on the thing here somewhere, so I can just show you the picture, because I haven't shown you the picture, but it's here. Bear with me, callers. Is it here? Ta-ta-ta. I wanted to show you the picture basically of us managing. There we go. There it is. I've got it now. I'm going to share my screen again. So this is Mr. Swan looking after our piles. We've got 4 windrows of human scale, and we're managing them by hand. This is Frida and Paula in the background. So we do a lot to it. We have collected a fair number of tricks of what we've actually added to this material, how we manage it, how we look after it. So there's a lot of work that's gone into it, and we're hoping that by the end of September there's going to be enough biology in it for us to extract it in good enough numbers to ship to you, so that you can drill your winter crops. In terms of, you know, some other things, this is that tent experiment that Adam was talking about. So again, we terminated this early, but to the left was the treatment, to the right was the control. No nitrogen used on either side, and we've already started seeing on the DNA analysis that it moves the needle.
Well, for that we used pretty terrible seed as well. So it was a good test of germination rates. We tested it in the lab, just in a paper towel. We got 100% germination rates, but the seed was, I guess it was identified as being low quality by whoever.
Reject. Yeah, it was.
Yeah, it was rejected seed. So yeah, so we obviously saw better germination and establishment of that seed, despite the fact that it was, you know, reject seed. We saw, yeah. Yeah,
perfect. And then, you know, as I followed up my my sand tomatoes with my airsoft BB gun pellets plants. So this is literally just a little corn flour growing out of a mixture of plastic balls and our Goop, and nothing else. So so there's definitely potential in this material, and you know, this is what it looks like for those of you who haven't had their hands on this yet. But it is usually somewhere between ketchup and fudge consistency. So we either call it the forbidden chocolate mousse or the forbidden chocolate fudge. If anybody's got a better name that's more marketable, come in and I'll definitely be your friend.
We're very keen on hitting consistent numbers. So at the moment, that's why the texture is kind of variable to hit those numbers. Obviously, as we get more money in and more research and development done, we can most likely make that a more consistent product in how it looks.
So yeah, so there's more pictures here, because this is what, you know, for instance, the carrots look like in the little trial, and you know, there was a 19% difference in the weight of carrots.
That's just one treatment to
just one treatment. Exactly, nothing else. You know, there's another picture of the sand tomatoes. So you yeah, we encourage your questions and we encourage your purchases so that we can actually get this into your hands and get it into the real world. Now there's a question in the chat. Oh, Clyde is going. Thank you very much for coming. Any more questions before we start wrapping up? I think we're good. Thank you very much for coming. This was recorded, so so we're going to post it on our channel and send to everybody else for review. Thanks, and we'll be in touch. And you can call us, email us, and we can answer your questions that way. So thank you very much for coming.
Thanks a lot, Daniel.
Cheers, guys.
Thank you.
Bye.